In the 19th century, people started dying in damp homes. Their rooms smelled faintly of garlic, and they shared one specific decorative trend: vibrant green wallpaper, dyed with heavy doses of arsenic. When Napoleon Bonaparte died in exile at Longwood House, his hair contained massive amounts of arsenic. He lived in damp, moldy rooms lined with that exact same green pigment. For decades, the pigment took the blame. But in 1890, an Italian physician isolated the real mechanism. A fungus called Scopulariopsis brevicaulis was living on the walls, feeding directly on the starch in the wallpaper paste. As the fungus digested the paste, it absorbed the inorganic arsenic from the dye and processed it into a highly toxic, volatile vapor known as Gosio gas. We had identified a microscopic organism whose basic metabolic survival routine quietly turned human living rooms into lethal gas chambers. Today, this fungus hasn't vanished with Victorian decor. It is an opportunistic pathogen that likely lives in the soil right outside your house. Its main target in humans is keratin, the tough protein that makes up our hair and nails. The fungal threads burrow into the nail bed, physically cracking and destroying the tissue to cause deep, stubborn infections. If it reaches a patient with a weakened immune system, it crosses into the bloodstream and invades deep tissues like the heart, lungs, and brain. The aggressive enzymes it uses to chew through human tissue are the exact same tools it used to digest Victorian wallpaper paste. It survives by consuming nearly any organic structure in its path, which brings up a serious biochemical problem. How does a single organism survive toxic heavy metals while also shrugging off our most advanced antifungal medicines? Through the Challenger pathway, the fungus absorbs solid arsenic, adds carbon and hydrogen groups, and turns the poison into a breathable vapor it exhales. This chemical mastery aids hospital survival. Efflux pumps recognize and actively force out modern antifungal drugs before they do damage. It doesn't invade human tissues out of malice. It possesses a highly evolved internal chemistry lab that neutralizes almost any external threat. To understand the true potential of that chemistry lab, we have to look far away from hospital wards, specifically to the Mediterranean Sea. Researchers investigating these marine ecosystems pulled up a deep-sea sponge and found something strange living inside its tissues. It was a unique strain of Scopulariopsis brevicaulis, known as LF580, thriving in an extremely salty underwater environment. When scientists sequenced its genome, mapping out more than 16,000 individual genes, they found hidden biosynthetic gene clusters. These are sections of DNA that code for complex molecules, but they remain completely silent when the fungus grows in normal dirt. The extreme marine stress activated two specific genes, NRPS1 and PKS2. Together, these genes act like a microscopic assembly line, combining amino acids and carbon chains to manufacture highly complex chemical compounds called scopularides. By forcing the organism to survive in a strange new environment, researchers proved its defensive chemical toolkit could synthesize entirely new biological molecules. When tested in the lab, these newly discovered scopularides did something remarkable. They actively inhibited the growth of human colon and pancreatic tumor cells. And scientists are finding uses for its aggressive digestion, too. The same powerful enzymes the fungus uses to break down wallpaper and nails can be deployed to clean up the environment. It readily consumes complex polycyclic aromatic hydrocarbons, the toxic chemicals left behind by crude oil spills. The fungus physically envelops the oil, pulls apart the complex ring structures, and digests the pollution. Its ability to withstand toxic chemicals, to reject modern drugs, and digest resilient materials made it a lethal pathogen. Now, those exact same traits make it a powerful asset for treating cancers and cleaning up petrochemical disasters. Scopulariopsis brevicaulis has traveled a strange evolutionary path. It has moved from the damp, arsenic-laced walls of old Victorian manors into the sterile wards of modern hospitals, and even deep down into the dense tissues of Mediterranean sea sponges. It reminds us that nature doesn't engineer life to be purely destructive or purely beneficial. It simply builds highly adaptable chemical toolkits designed to ensure survival under extreme stress. By learning how to read its genetic code and manipulate its environment, we have taken a microorganism that spent the 19th century poisoning us and turned it into the medicine and the environmental cleanup crew of the future.